All right, Team Malolos, at this juncture, we will start discussing Module 2. And as we said, ang Module 2, this talks about drug targets. In this regard, we are going to discuss the different macromolecular targets by drugs for them to produce effects. Recall yesterday that I introduced that in general, we have two major macromolecular targets brought about by drugs. Those are proteins. I hope you could still remember that. And also nucleic acids. Between nucleic acids and proteins, majority of drugs, they do target proteins. That's why in the interest of time, we will focus on drug targets, particularly proteins. Okay? Kaya magfo-focus tayo sa proteins. Let me remind you that the knowledge that you obtained or acquired during biochemistry days, gamit na gamit sila sa module na to. Kaya I hope meron kayong baon-baon na kahit papaanong stock knowledge sa biochemistry. Okay? For the key concept, general concept about drug targets, particularly proteins, as I've said, majority of the pharmacological agents that are available in the market or yung mga dinidiscover pa lang or pinag-aaralan pa lang, they target proteins. This is easy to comprehend because majority of macromolecules present in our body that have functions, proteins, kay marami kasi. Examples are receptors, enzymes, and even transport proteins. Do not worry because later we will discuss them individually. Therefore, it is important to know their general functions for us to appreciate the actions of drugs. Because basically, as I've introduced yesterday, ang major role ng gamot is to modify the function, to alter the function, to change what is happening inside the body brought about by proteins. Kaya kailangan malaman nyo muna kung ano ba ang usual functions nila. Do you understand? Am I clear to everyone? Okay, nice. So that's why we are going to review some concepts about proteins. As you learned during biochemistry days, medyo fresh ito kasi last semester lang. Proteins are considered as the most abundant macromolecules and they perform a wide array of functions. And the basic functional unit or the building blocks of proteins are the amino acids, right? Okay, mga amino acids. And when we talk of amino acids, for them to be polymerized into proteins, they should form peptide bonds. And kapag nag-form sila ng proteins, they will be functional if they will undergo folding process. Okay? They will undergo folding process. So siguro na-encounter nyo na to ng biochem. Ang point lang yan is lower level of macromolecules, amino acids. And then pag nag-ipon-ipon sila, they do fold, becoming now complex proteins. That's why they are considered to be functional. Okay? In relation to that, para makita natin ang hierarchy ng folding process ng mga amino acids into complex proteins, we should recall the different protein structures. So remember during biochemistry that there are different hierarchy of protein structures that would tell us if sila ba ay non-functional pa or functional na. Okay? Kung sila ba ay may pakinabang na sa katawan or meron na silang specific roles in the body. Natatandaan nyo ba or familiar ba kayo sa biochem sa primary, secondary, tertiary, and quaternary structure? Yes or no? Very good. So it's a yes. Siyempre, last sem lang yan. So we have some recall questions or recall concepts about the levels of protein structure. Number one, we have the primary structure. Basically, nabanggit ito before na ang primary structure, this is comprised of polypeptide chain. Diba? This is comprised of polypeptide chain. Ay polypeptide chain, ibig sabihin, dikit-dikit na amino acid sequences that are being connected by peptide bond. Okay? Being connected by peptide bond. So yun yung primary structure. And then, when some rotational bonds become near with each other, they do fold and hydrogen bond will be formed. Eventually, kapag nagkaroon ng folding through the presence of hydrogen bonds, that's the time that secondary structure would emerge or would arise. So we have two common types of secondary structures. Those are the alpha helices or alpha helix and the beta pleated sheets or pleated sheet. So the picture found here is an example of an alpha helix. Okay, familiar na kayo dyan. And also the beta pleated sheet. Ang point lang yan, kaya nagiging ganyan ang itsura ng mga secondary structures because of hydrogen bonds. Okay, because of hydrogen bonds. Are you ready sa tertiary structure? Hmm? Okay, moving on, we have the tertiary structure. Mind you, class, that tertiary structure is the proof or evidence that the protein is already functional. Kapag na-reach ng protein ang kanyang tertiary level of structure, that's the time na siya ay functional na. In biochemistry, if you are not familiar, the technical term if a protein is already functional, that is native conformation na siya. Okay? Native conformation. So kapag functional, it's called native. Now, balik tayo sa primary kasi nakalimutan kong sabihin. Sa primary, nabanggit natin na polypeptide chain siya in which proteins are not yet functional. At ang tawag natin sa non-functional form ng protein at its primary structure is nascent polypeptide. Nascent. Okay? Familiar ba sa terms nascent or nascent and native? Or newly heard so far? Pasensya na uminom ako kape from time to time. Okay, very good, familiar. So polypeptide chain is nascent and then, primary structure nascent. Pag tertiary level na, yan na yung native conformation. Okay? So ang tawag natin sa isang polypeptide, isang pirasong polypeptide chain na naging functional ay isang subunit. Now, in some cases, there are proteins na more than one subunits ang composition for them to become functional. So ang tawag na doon ay quaternary structure. So more than one na siya. More than one subunits na para maging functional. Kaya makikita nyo, ilang piraso ang nakapresent sa slide kasi it requires more than one. Okay? More than one subunits para maging functional. Okay? In other words, ang difference or similarity, again ulitin ko, ang similarity ng tertiary at quaternary, functional na yung proteins. Kaya lang ang pagkakaiba nila, kapag tertiary, isa lang na chain ang naging functional. Okay? Or functional na siya at one chain. Pero pag quaternary, it requires more than one. Okay? Anong example niyan para ma-internalize nyo mabuti? Ang example, kapag tertiary, myoglobin. Siguro narinig nyo na to before sa biochem. Okay? Myoglobin kapag tertiary. Hanggang tertiary lang siya kasi isang polypeptide chain lang siya. Pero kapag quaternary, hemoglobin na yon. Kasi ang hemoglobin, 4 na subunits ang kailangan. Okay? Ano lang naman to? Review. Okay? So that's our review regarding protein structures or conformations. So take note, kailangan mag-fold ng proteins for them to be functional. Next, pwede na tayo sa susunod na slide. Okay, very good. Next, let's also note that once proteins were synthesized, remember in biochem also that the process of protein production is called translation. Okay, that's called translation. Of course, after translation, there are still modifications being done so that madadiversify pa ang function ng protein sa katawan natin. Ang tawag natin sa modifications na ginagawa ng katawan sa mga proteins na na-produce ay post-translational modifications. So in that case, let me give you examples. Okay? Let me give you example post-translational modifications. Number one, in the case of collagen. Collagen is a protein also. That is a fibrous protein. Makikita siya sa ating mga blood vessels, sa ating mga cartilage, sa skin. In other words, abundant fibrous protein siya. Okay? Ang collagen ay matibay na protein. And ang important component ng collagen para sa kanyang strength, ay ang isang amino acid called hydroxyproline. Okay, that is called hydroxyproline. Ulitin natin, important component ng collagen to make it strong. Okay? It provides strength, that is hydroxyproline. However, as you can tell, hydroxyproline is not a standard amino acid. Derived na siya. May pinanggagalingan siya, which is proline. Okay, which is proline. Kaya yung picture dito na makikita nyo, that is the original amino acid, the standard amino acid proline. So para makagawa tayo ng hydroxyproline that would provide strength, this proline has to be modified. Kaya ang tawag doon, post-translational modification. Did you get that? Can you follow? What about the others? Mangilan-ngilan yung commonly sumasagot. Okay. So, very good. So, i-continue natin. Paano mamomodify ang proline? It has to undergo a chemical reaction. So, ang tawag natin sa chemical reaction is hydroxylation, forming now hydroxyproline. Okay, forming now hydroxyproline. And in connection to that, let me emphasize that in hydroxylation reaction, we need a cofactor such as vitamin C. Okay? Kailangan natin ng supply ng vitamin C para makapag-produce na maraming hydroxyproline. Okay? The more vitamin C that we have, more hydroxyproline. Okay? Mamaya, babalikan natin yung reflection dyan. May reflection tayo dyan mamaya. I'm just discussing right now the general function. Okay? Next, dun tayo sa second example ng post-translational modification. Next stop, we have prothrombin. Okay, we have prothrombin. Ang prothrombin ay isang halimbawa ng clotting factor. Okay, isa siyang halimbawa ng clotting factor. In other words, this is a type of protein that is needed for clot, okay, for clotting or pamumuo ng dugo. All right? Now, for this prothrombin or clotting factor to have its specific function in causing clot, it must undergo modification as well, that is via the chemical reaction carboxylation. Okay, carboxylation. So in this case, once na carboxylate siya, that becomes biologically available for action. So yun yung function ng prothrombin kapag siya ay undergo ng carboxylation. So nagkakaroon ng clotting. Okay? O sulat natin sa taas strength ng collagen. Okay? Nagpo-provide ng strength. So yan po ang dalawang examples ng post-translational modification. Again, I'm just discussing right now general function. Mamaya i-connect natin ang drugs. I-connect natin ang drugs. Now, type done if you're now ready to apply the actions of drugs here. Okay, very good. So makinig mabuti makinig mabuti. Sir, anong essence ng pagbibigay mo na example ng mga common processes ng post-translational modifications? As I've said, for you to discover drugs as medicinal chemists, when discovering drugs, bago sila maghanap ng gamot, bago sila maghanap na magmo-modify ng function, they should know the normal function. These are normal processes in the body. These are physiological in the body. Okay? So once they determine this, they could think of a solution or a drug that could alter this function. Okay? Let me give you an example. Okay? Unahin natin yung sa clotting. Okay? Clotting. Doon tayo lahat sa clotting factor. Guys, kapag nagkaroon ng abnormality sa clotting sa ating katawan, lalong-lalo na yung mga at-risk patients like, for example, may cardiovascular diseases, may chance na kapag nagkaroon ng spontaneous clotting, they may suffer from stroke. Tama? They may suffer from stroke. And also, myocardial infarction. So one of the problems kapag may abnormal clotting sa mga vulnerable patients, they may suffer from stroke and also MI. Kaya nakita ito ng mga scientists as problems at nakita nila na ang isang mechanism leading to this is carboxylation reaction. So anong ginawa ng mga medicinal chemists? They search for drugs that could target carboxylation reaction, okay, that could target carboxylation reaction so as to stop formation of prothrombin needed for clotting. Did you get what I'm saying? Naiintindihan nyo? Hmm? Naintindihan nyo ba yung sinasabi ko? Okay, that's why na-discover nila ang isang gamot na nagbablock ng carboxylation. That is actually called warfarin. Narinig nyo na yung warfarin? Familiar sa warfarin? May ibang familiar, may ibang hindi. Ang warfarin ay isang blood thinner. Okay, blood thinner, ibig sabihin pinipigilan niya ang pamumuo ng dugo. So ang mechanism of action ng warfarin, ito ay general mo wala nga, may mas malalim na explanation dito, pero binibigyan ko lang kayo ng idea anong importance ng pag-alam ng normal happening in the body at kailan papasok ang action ng gamot. So in that case, we have warfarin that can block carboxylation reaction. So in that case, I have some questions. Kapag na-block ng gamot na warfarin ang carboxylation, meron ba tayong carboxylated prothrombin? Meron o wala? None. Kapag wala, sa tingin nyo ba mai-stop o mape-prevent yung clotting? Yes or no? Yes. So pag walang namuong dugo, may magbabara ba sa ugat? Yes or no? Meron ba o walang magbabara sa ugat? Wala. Kaya napipigilan ang stroke at MI. Kaya ginagamit to ng mga patients na may na at risk sa stroke and MI, lalo na kapag may history sila ng spontaneous clotting. Na-appreciate nyo yung pagpasok natin sa drugs doon sa process ng modification? Okay, so let's continue. Tuloy natin. Doon tayo sa taas, sa hydroxyproline. Nung nalaman na ang hydroxyproline ay napoproduce because of hydroxylation at sinusuportahan ang vitamin C, sinusuportahan ng vitamin C ang prosesong ito, naisip na mga businessmen, actually ang mga pharmaceutical companies, business yan, okay? Pera-pera lang din talaga sila. Kaya nakaisip sila ng paraan para i-market ang vitamin C. Alam niyo yung potency ni Bea Alonso? Anong pansin niyo sa strategy nila Bea Alonso nung minamarket ang potency? 'Di ba sabi pampaganda ng balat, pampaganda, yes, pampaganda. Totoo 'yon, hindi lang siya immunity. Kasi kapag ikaw ay umiinom ng vitamin C, marami kang vitamin C supply sa katawan na susuportahan mo ang hydroxylation, ang collagen sa balat mo tumitibay, nababanat siya. So pampaganda ng balat talaga ang vitamin C. Kaya may sense yung marketing nila Bea Alonso. It has something to do with hydroxylation reaction. So the next time na uminom ka ng vitamin C, pwede mo nang explain sa mga kasama mo sa bahay na dumadami ang hydroxyproline that provides tensile strength sa collagen, gumaganda po ang balat natin. So yon. Gets nyo ba? Okay. So yun yung importance. Actually, na-explain ko na yung slide na to. O move on na tayo. So ang importance is once you know the usual process, you could identify a gap and solve it. Okay? Ganun lang siya kasimple. So in that case, pwede na nating palalimin pa ang discussion sa mga potential drug targets. So ngayon, lumalalim na ang understanding natin sa salitang target. Ito talaga yung pupuntahan ng gamot, thus modifying the function. Okay? So since, as I've said, ang focus natin ay proteins, unahin nating i-discuss ang mga proteins such as structural proteins. So for you to appreciate discovery of drugs that target structural proteins, let me give you some insights first about structural proteins. What are these? Ang structural proteins, these are macromolecules which are important, okay, which are important for mitosis, ay important sila for mitosis. So when we say mitosis, anong natutunan niyo sa mitosis? Anong nagdi-divide sa mitosis? Cells. Nice. Okay. So important ang structural proteins for cell division. And aside from cell division, important din ang structural proteins for cell movement. Okay? Cell movement. In particular, applicable ito sa mga inflammatory cells. Okay? Applicable ito sa mga inflammatory cells like neutrophils, okay? Like neutrophils. So when we say cell movement, gagalaw ang cell from one location to another, such as in the case of inflammatory cells, neutrophils. So when I say cell movement, halimbawa, susugod sila kung saang area sila kailangan. Nakakarating sila doon kasi they have structural proteins. And aside from that, axonal movement, axonal movement of neurotransmitters. And then we have axonal movement of neurotransmitters. So ito po yung mga functions ng structural proteins sa ating katawan. Okay? Ano ba yung mga examples ng structural proteins in the body? Ang example natin ng structural proteins, tubulins. Tubulins in which kapag sila ay nag-ipon-ipon or nag-polymerize. P here stands for polymerize. Pag sila ay nagdikitan, they become microtubules. Okay, they become microtubules. Have you heard the term microtubules dati? Common term siya sa cells, di ba? Okay, so nice. So example natin, we have tubulins na pag nag-undergo ng polymerization, they become microtubules. So thanks to them, may cell division, may movement of inflammatory cells, at may axonal movement. Okay? Pwede natin continue mga bata. Okay, nice. So what's the importance of mentioning these functions? In some situations, these processes are said to be bad or adverse in the body. Masama, okay? Let me give you an insight. Halimbawa, if a patient has cancer, okay? If a patient has cancer, cancer is characterized by abnormal cell division, okay? Abnormal cell division. Therefore, pag may abnormal cell division, masyadong tumataas ang production ng mga cancer cells or dumarami ang production ng mga cancer daughter cells or daughter cancer cells. Okay? So yun yung nangyayari. Masyadong dumadami ang mga cancer cells. Now, one of the possible suspects ay one of the possible reasons kung bakit nagkakaroon ng abnormal number of cancer cells, abnormally functioning ang microtubules and tubulins. That's why in cancer, masyado ring enhanced ang mitotic process. Okay? Masyadong enhanced or abnormal ang cell division caused by tubulins and microtubules. So with that being said, since abnormally functioning ang tubulins at microtubules, question sa inyo guys, a question for you. Do you think if we want to target cancer, if we want to treat cancer, pwede bang drug target ang mga tubulins at microtubules? Yes or no? What do we want sa mga tubulins and microtubules? Do we want to support their action or block their action para ma-stop ang cancer? Block, very good. Kaya meron tayo merong diniscover na drugs ang mga medicinal chemists na ang role ay i-block ang polymerization ng tubulins. For example, pag sinabing i-block ang polymerization ng tubulins, walang microtubules. Pag walang microtubules, walang mitosis. Kaya meron tayong currently available anti-cancer vincristine. You know vincristine, familiar? Mga vinca-alkaloids? Narinig na ang vincristine? Hindi pa. Okay. So vincristine, ito yung mga vinca-alkaloids kung tawagin. Burahin nyo itong parenthesis ha. Examples, you have vincristine, vinblastine. Okay? Ito yung mga anti-cancer agents that can block mitosis because they inhibit polymerization of tubulins. Okay? They inhibit polymerization of tubulins. What else? Aside from vincristine and vinblastine, we also have, siguro familiar kayo dito, paclitaxel. Narinig niyo na yung paclitaxel? Or not yet? Okay lang, basta honest kayo, no problem. Hindi rin. So at least narinig nyo na sila. So we also have paclitaxel. Pag sinearch nyo sa internet, anti-cancer din sila. Okay, anti-cancer. Ito yung mga anti-cancer agents that can target mitosis. Are you learning so far, guys? Okay, continue natin. Ito, pusta ko, familiar kayo dito. Colchicine. Narinig nyo na yung colchicine? Ayan, familiar kayo dyan, colchicine. Can you tell me, saan ginagamit ang colchicine or anong use ng colchicine? Yes, very good. Somebody answered it. Yes, that's nice. Ang colchicine ay anti-gout. Okay? Anti-gout ang colchicine. That's right. Okay, that's right. Masakit ba ang gout? Makirot ba ang gout? Yes. Ang problem sa gout, para lang may idea kayo ha, ano to, para lang ma-appreciate nyo kung ba't nagdi-discover ng drugs. Okay? Ang gout ay isang inflammatory disease caused by accumulation of uric acid or urate crystals sa ating joints, sa joints. So kapag maraming uric acid or urate crystals sa ating joints, ang tendency na-attract niya ang mga neutrophils, makinig na-attract niya ang mga neutrophils to attack the joint. Neutrophils can cause pain and inflammation in the joint. Kasi nga, because of high urate crystals, na-enhance ang movement ng neutrophils. Na-enhance ang movement. Pag maraming neutrophils doon sa joint na may uric acid crystals, may inflammation, makirot siya. Okay? Now, anong role ng colchicine? Ang colchicine, ang gagawin niya, ibablock din niya ang polymerization ng tubulins. So pag na-block ang polymerization, meron o walang microtubules? Meron or wala? Very good. Walang microtubules. Anong importance pag binlock mo ang microtubules? Walang movement of neutrophils. Napipilayan ang neutrophils. Kaya pag napilayan ang neutrophils, hindi sila makarating sa joint, hindi sila makapag-cause ng inflammation, nawawala yung kirot sa joint. Kaya ang colchicine ay anti-gout. Naintindihan? Okay. So yan, may molecular reason yan pero I just want you to have an idea. Okay? I just want you to have an idea. So those are the currently available drugs that can target structural proteins. O hindi natin alam, baka isa sa inyo, pag naging scientist siya, maka-discover pa ng ibang gamot na pwedeng maging anti-gout, na pwedeng maging anti-cancer, ang goal niya, targetin ang structural proteins. So yon, kaya kailangan alam nyo ang function ng mga structural proteins. Are you learning? Okay, so let's continue. So dito naman tayo sa second, or review tayo ha. Ang unang protein na na-discuss natin, structural. Moving on, we have transport proteins. Transport proteins, these are, define muna natin siya. Tinamad si teacher mag-type kung anong definition, sulat ko na lang. Ang transport proteins, these are cell membrane proteins. As you can see, nasa cell membrane sila naka-embed. Okay? These are cell membrane proteins that undergo conformational changes, okay, that undergo conformational conformational changes kapag may nag-bind sa kanilang mga substances. Okay? So these are cell membrane proteins with specific binding sites that undergo conformational changes. What do we mean by that? Pag sinabi nating nag-undergo ng conformational changes, pag dumadaan ng isang compound sa kanila, nagbabago sila ng itsura o ng shape. So for instance, general illustration tayo ng transport protein. For example, we have here the cell membrane, and then yung malaking structure that you can see here highlighted in color yellow, that is a transport protein. So it allows movement of compounds such as polar molecules para makapasok sa loob ng ating cell. Okay, para makapasok. Now, kapag dadaan ang isang compound via the transport protein, pag-bind niya sa active site, kay pag-bind niya, palitan natin ng color, pag-bind niya sa active site, ang tendency, transport protein will change its conformation. In other words, it changes its shape. Paano nag-change ng shape? Yung active site, open siya sa labas nung una. Due to its changes in conformation, sa loob na siya naka-open, allowing entry of that polar molecule. So ganun lang kasimple ang ibig sabihin ng transport protein. Nagbabago ng shape to allow movement of compounds. Gets? Okay, so proceed tayo sa example para ma-appreciate nyo ang drugs. Ang example natin ng transport protein ay yung potassium ion and proton pump. In short, ang nickname nito ay proton pump. Okay, proton pump. I repeat, example of a transport protein is the proton pump. Proton pump is found in the stomach. Okay? It can be found in the stomach. Particularly, it can be seen, again, it can be seen in the parietal cells of the stomach. Again, it can be seen in the parietal cells of the stomach. So anong role? Anong role ng proton pump? To make this short at mas madaling maintindihan, it allows exchange of potassium and also proton. Ganun lang kasimple. May exchange ng potassium. Tingin sa PowerPoint, wala mo na magsusulat. Wala mo na magsusulat. Tingin sa PowerPoint, ay makinig. Ang proton pump, it allows exchange of potassium being encircled and also proton. Paano ang exchange ng proton at potassium dito? Ang proton, makinig, ang proton ay lalabas mula sa parietal cells. Ay lalabas mula sa parietal cells, kaya makikita na natin siya na na-release sa stomach. So napapataas ng proton pump ang proton sa stomach natin. Okay? Napapataas ng proton pump ang proton sa ating stomach. Okay? Nare-release kasi niya. And in addition, once secreted ang proton, it will react with chloride, forming ultimately hydrochloric acid. Forming ultimately hydrochloric acid. Gets? Ha? Gets? Okay. So with that being said, let's try to reflect on it. I-reflect natin. Kapag active ang proton pump, taas o baba ang proton sa ating stomach na nare-release? Taas or baba? Taas. When reacted with chloride, na-form ang hydrochloric acid. Therefore, pag so much active ang proton pump, taas o baba ang hydrochloric acid sa stomach natin? Answer. Taas. Very good. Taas. Ano ang hydrochloric acid? Acidic or basic? Acidic. Very good. Pag sumusobra ang hydrochloric acid sa stomach, mahapdi ba? Mahapdi ba? Yes. Kaya if there is so much hydrochloric acid, masyadong marami ang acidic compound, kaya may hyperacidity. Masakit ba to? Sakit ba to, rather? Sakit ba siya? Yes. So kung sakit siya, problema ba siya? Yes. So therefore, pwede bang i-eye ng medicinal chemist ang proton pump para mapigilan ng hyperacidity? Yes. Kaya meron tayong currently available drugs that can block proton pump. And these are the proton pump inhibitors. Familiar ba sa omeprazole halimbawa? Yes. So ito yung mga proton pump. You have Google. So search nyo na lang ano yung mga examples ng proton pump inhibitors. Binibigyan ko lang nga kayo ng idea anong importance ng drug targets. Okay? Gets? So omeprazole, esomeprazole, rabeprazole, yan yung mga proton pump inhibitors. Type done if you're ready sa next slide. Okay. So far, nakakailan na tayong proteins na nadidiscuss? Dalawa. Very good. Tip ko sa inyo pag nag-aral kayo o pag nakikinig kayo, lagi nyo reviewin yung mga dating na-discuss kasi mahirap kasi ang mahirap sa estudyante minsan, segmented nagigets nila. Ibig sabihin ng segmented, slide 1 na gets, slide 2 na gets, slide 3 na gets. Pero pag sinamarize lahat, hindi na na-gets kasi hindi nila kino-connect sa mga nakaraang slide. Kaya whenever na nagdi-discuss ako, lagi kong sinasabi, ano na na-discuss natin para ma-reinforce yung long-term memory nyo. Okay? Kaya lagi tayo nagre-review doon. So anyway, proceed tayo sa number 3, enzymes. Ang enzymes na-discuss na sa biochem. Kaya medyo shortcutin natin ito. I-reinforce na lang natin yung mga na-discuss about enzymes. Ang enzymes, these are so-called as biological catalysts. Okay? These are so-called as biological catalysts. So this means that enzymes can hasten chemical reactions. So when we say hasten chemical reaction, pinabibilis nila ang chemical reaction. Okay? That is because enzymes have the ability to lower the activation energy. Okay? They lower activation energy or Ka. Familiar ba sa term na activation energy? So siguro familiar kayo doon because you had this in biochem. So enzymes can lower activation energy. So for example, sa figure, sir, ano ba yung activation energy? So halimbawa sa figure na ito, para hindi kayo malito, hatiin natin ito. Ay, hatiin natin. Number one, sa left side, without enzyme, walang enzyme. Okay? Chemical reaction without the enzyme and chemical reaction with the enzyme. As you can see, dito sa chemical reaction na wala pang enzyme, walang enzyme, ang activation energy is measured at this level. Okay? So ito yung level ng activation energy without the enzyme. So anong realization pag ganyan ang level? Okay, ganyan ang level. Medyo matagal yung chemical reaction bago matapos. Okay, medyo matagal. Or mabagal, yes, that's right. However, sa right side, you can see with enzyme, with the introduction of enzyme, bumaba yung transition state. So pag sinabing bumaba ang transition state, ang activation energy is measured at this level na lang. So makikita natin na ang activation energy as compared sa walang enzyme, bumaba siya. And mababang activation energy would tell us that the reaction happened faster. Okay? Kaya marami tayong mga products in the body na nafo-form ng medyo mabilis kasi kasali ang enzyme. Okay? Nagpa-participate ang enzyme. Okay, so yan ang kayang gawin ng enzyme. They can participate sa chemical reaction to make it happen or to make it go faster. Okay, so yan yung function ng enzyme. Now, paano gumagalaw ang enzyme? Basically, ganito ang ruling sa enzyme. Ay dito tayo sa kakadagdag ko lang nito kagabi. Picturean nyo na lang after kong i-discuss. Makinig muna sa akin. Nakikinig pa ba? Okay, nice. So take note, ganito kasimple ang ruling sa enzyme. Okay? Ang enzyme para siyang general chemistry na kapag may chemical reaction, di ba? Reactant, via a chemical reaction, nagiging product. Okay, nagiging product. Now, sa biochem, nagkakaroon lang ng konting pagbabago sa naming. Okay? So ang reactant sa biochem, nagiging substrate. Okay, nagiging substrate. So isipin nyo lagi pag substrate reactant yon. Okay, yun yung babaguhin. And then, yung chemical reaction nangyayari kasi may participation na ng enzyme. May participation ng enzyme. And because of enzyme, yung substrate, product pa rin ang tawag. Okay, product pa rin ang tawag. Nabubuo ang product ng chemical reaction or enzymic reaction ng mas mabilis. Naintindihan? Naintindihan general concept? Okay, so that's why in the figure shown, we have here the substrate. This substrate or the reactant, okay, para siya ay mag-undergo ng chemical reaction, it should bind to the enzyme's active site. Ang active site, ito yung butas or pocket na pupuntahan ng substrate. And then, pag nagdikit na ang enzyme at substrate, may chemical reaction na forming now a product. Okay, forming now a product. Okay, so ganyan yung function ng enzyme. Pwede na dito, guys? Sorry, pipicturean nyo pala. Sige, picturean nyo na, go. Type done if you're finished taking a picture or screenshot siguro ginawa nyo. Sige, type done na lang ko, okay na. Okay, so next. Punta na tayo sa slide na to. So heads up lang guys, heads up. Bakit natin sinasabi ang action ng enzymes? Guys, napakaraming chemical reactions sa ating katawan being mediated by enzymes. So pag maraming chemical reactions, maraming mga products tayo ma-observe sa katawan. And some products could be bad to us. Kaya to be able to address that problem, we need to interfere the chemical reaction by targeting the activity of enzymes. Okay, let me give you an example. Isa lang, pero napakadaming examples ito. Kayo nang bahala makadiscover sa buong journey nyo sa pharma life. Okay? So example, we have the enzyme alpha-glucosidase. So explain natin ang function ng alpha-glucosidase. Ang alpha-glucosidase ay isang enzyme. Ito siya, i-highlight natin. Alpha-glucosidase. Alpha-glucosidase is an enzyme that can convert oligosaccharides into monosaccharides. Okay? So this enzyme can convert oligosaccharides to monosaccharides. Question, what's the difference between oligo and monosaccharides? Take note, ganito lang siya kasimple. Compared to monosaccharides, ang oligosaccharides ay complex carbohydrate pa siya. Okay? Sorry. Complex carbohydrate pa siya. Okay? Complex carbohydrate. While ang monosaccharide, di ba pag mono isa lang, so simple form na to. Okay? Simple form ng carbohydrate. I repeat, ang difference between oligo and monosaccharide, ang oligo ay complex carbohydrate at ang monosaccharide ay simple carbohydrate. In other words, ang role ng alpha-glucosidase, pasimplihin ang structure ng sugar. Pasimplihin ang structure. Question, ano overall ang importance, bakit pinapasimple ng alpha-glucosidase ang sugar or carbohydrate structure? Nakikinig pa ba? Ang question ko is, anong importance? Ano ang kahalagahan ng pagsisimplify ng structure? Ito ang importance niyan, makinig. Sa katawan kasi natin, pag kumakain tayo ng pagkain, kapag ang carbohydrate na nakain natin ay complex pa ang structure, non-absorbable pa to. Hindi to kayang ipasok sa sistema ng katawan. Okay? Hindi kaya. So yung sugar form na complex, hindi siya makapasok sa katawan natin, hindi siya mapakinabangan. Okay? Hindi makapasok sa bloodstream. Okay? Ang naa-absorb, ang absorbable sa ating small intestine para makapasok sa systemic circulation, yung simple form which is monosaccharide. Kaya makikita nyo sa ating dugo, glucose, kasi yun yung absorbable. Gets nyo? Yung difference ng complex at simple? Yes. O bakit nasisimplify? Sinong enzyme ang kailangan? Alpha-glucosidase. Continue. Continue natin. Okay, very good. Now, syempre, kapag ang alpha-glucosidase ay ina-eye din, tina-target din ng mga medicinal chemists. Why? Let me give you an instance. What if merong enhanced activity ng alpha-glucosidase? Pag may enhanced activity ng alpha-glucosidase, less or more glucose absorbed? Go. Anong answer? More. Very good. More glucose being absorbed. Question, pag more glucose being absorbed, taas o baba ang blood glucose? Very good. Tumataas ang blood glucose. Meron bang sakit na pwedeng i-associate sa increased blood glucose? Yes, what is it? Yes, very good. So that is diabetes mellitus. Okay, diabetes mellitus. So ang diabetes mellitus ay isang condition characterized by increased blood sugar or blood glucose level. So i-connect natin yung enzyme, makinig na i-connect natin yung enzyme na alpha-glucosidase sa diabetes mellitus, kaya pwede mong i-eye as target ang alpha-glucosidase. Kasi kapag tinarget mo ang alpha-glucosidase, say for example, if you did block, pag binlock mo ang alpha-glucosidase, less glucose absorbed, less blood sugar, anti-diabetic effect. Kaya meron tayo mga gamot called alpha-glucosidase inhibitors. So anong realization natin sa mga alpha-glucosidase inhibitors? Pwede ba silang i-categorize as anti-diabetic agents? Yes or no? Pag ang gamot ay alpha-glucosidase inhibitors, Sila ba ay anti-diabetic? Yes. Okay, very good. So, ang example niyan ay si acarbose. O type nyo sa Google, acarbose. Makikita nyo, anti-diabetic drug siya. Okay? We also have voglibose. Okay, voglibose. Yan talaga yung pangalan niya. Okay, so acarbose, you also have voglibose, and also miglitol. Kaya may gamot tayo na alpha-glucosidase inhibitors. Are you learning, class? Okay. Type done kung ready na sa susunod. Okay? Kasi ang susunod ata natin, yes, receptors. Okay. So next, recall tayo, nakakatatlo na tayo mga proteins na natatarget ng gamot. Inspiration ito sa inyo kapag nagthesis kayo halimbawa. Hopefully sa time nyo pa nagthesis kayo ay face-to-face na. Sana para hindi kayo malimit sa descriptive study. Kasi ngayon puro descriptive gawa ng hindi makapag laboratory eh. Kasi exciting yung thesis sa pharma kapag experimental research. Kumbaga itong subject na medchem, yung pagiging investigative nyo, magkakaroon kayo ng inspiration na mag-thesis kahit papano kasi nagkaka-idea kayo ano ba yung style. Lalo na sa module 3, yung drug discovery. Magkakaroon kayo ng idea paano ba dinidiscover yung gamot often. Okay? Pwede na po sa 4 receptors. Okay, so nice. So next, eto feeling ko challenging na yung part na to, pero let's see. I'll do my best to explain this accordingly para maintindihan nyo. So last, we have receptors. Let's define receptors first. A receptor is a functional macromolecular cell component with a specific stereochemical configuration where a ligand interacts usually in a lock and key fashion. Sir, ang haba ng definition. Wala kami naintindihan. Okay, one step at a time. I-highlight muna natin ang mga significant terms, yung mga keywords na makakatulong sa atin upang maintindihan ito. Gusto kong bilugan nyo or i-highlight nyo ang mga salitang ito. Number one, we have specific. Okay, number one, I want you to highlight the term specific. Another, we have ligand. And then we have lock and key fashion. So I want you to highlight these terms. This will help us out in understanding what are receptors. Okay, so isa-isahin natin. Number one is specific. Pag sinabi nating specific, ang receptor ay isang protein na maarte. Okay, maarte siya in a sense that it only allows binding of specific compounds. Yun lang, okay? Halimbawa, meron tayong receptor 1, meron siyang specific compound na tinatanggap lang. Okay, specific lang 'yon. Hindi siya mag-a-allow na pumunta ang iba mga components or ibang agents. Okay? So yun yung una, specific. Pangalawa, ligand. Ano ba yung ibig sabihin ng ligand? Ang ligand, ito kumbaga, analogy natin ito kumbaga yung mga candidates for binding. Ito yung mga compounds na may K or may kakayahan na mag-bind doon sa receptor's pocket or active site. Okay? Ano ba yung mga examples natin ng ligands? Actually, ang mga ligands na ito, yung mga candidates for binding, pwede itong endogenous, pwede itong exogenous. So pwede itong natural compounds, okay? Pwede itong mga natural compounds. I think na enumerate ko kahapon yung mga natural compounds that could interact with proteins such as receptors. Naalala niyo ba yung mga neurotransmitters, hormones, sila yon, okay? Sila yung mga natural messengers and otacoids. So we have neurotransmitters, hormones, and also we have otacoids. Okay? And then, of course, aside from natural, syempre, meron din tayong exogenous. So pag sinabi natin exogenous, pasok na dyan ang mga xenobiotics. Kasali ba ang drugs sa xenobiotics? Yes, okay. So ligands could be natural and then xenobiotics drugs. And finally, punta tayo sa lock and key. Again, kung nakikinig pa guys, we have the lock and key fashion. Lock and key is the analogy that explains the interaction between the receptor and the ligand. Wherein lock is the receptor, a lock is the receptor, and the one that could open that lock, is the key, the susi, which is the specific ligand that is complementary to the receptor, okay? Na matched doon sa receptor. So for you to understand this all together, let's try to analyze the figure shown below. Okay, let's analyze this. So for example, we have here the given receptor. Again, we have here the given receptor. Of course, it has an active site wherein not all messengers, not all ligands could bind. Syempre, specific lang because lock and key nga siya eh. Suppose a specific ligand, yung messenger dito siya rin yung ligand ha, it could be in the form of endogenous or synbiotic nga. So let's say this ligand is attracted to the receptor or matched sila. Once bound, that causes activation of the receptor. You can now open the lock, as we tell. Okay, you can now open the lock. And when I say open, ibig sabihin nun, meron tayong signaling mechanism. Okay, meron tayong signaling mechanism or signal transduction. Yung message na nakalagay dyan, signal transduction din yon. Okay? So signal transduction mechanism that would lead ultimately to an effect or physiological effect. So once this ligand is bound to the receptor, that causes messaging or signal transduction mechanism that would finally lead to cellular response or effect. So yan ang function ng receptor. Ayan yung function ng receptor. Pwede kong mag-continue? Okay, so let's have some reflection ulit dito sa receptor. Sir, ano pong importance na inexplain nyo in general ang ginagawa ng receptor? Ulitin natin, bawat receptor sa katawan may kayang gawing epekto sa cells. Okay? Kaya lang, hindi nila basta-basta nagagawa yon kasi wala silang susi. They need the key, they need the ligand for them to express that effect that they have. Okay? Kaya isang inaay na target ng mga medicinal chemists ang receptor kasi kapag nag-discover sila ng synbiotic or drug that could act as a key to the receptor, that drug could serve as a reason para ang receptor ilabas niya ang epektong meron siya. Kaya inaay itong target ng mga scientists pag nagdi-discover ng gamot. Naintindihan? Okay. Kaya ang challenge dito is paano ko magagawa ng susi sa maarting lock? Kasi ang lock nasa katawan na eh. Alam naman baguhin mo yung receptor, itsura na receptor sa katawan. You can never change that. Yun na talaga yung itsura nung lock. Kaya ang challenge is i-modify ang susi, ihinang ng ihinang, gawing fine ang susi para makapasok to, para ma-push ang pasok niya sa receptor, may effect. Okay? Ganon ang challenge dyan. Ngayon, para mas makilala natin ang mga cellular responses brought about by receptors, let's now proceed to the different types of receptors. Depending on the message that they relay, depending on the signal transduction that they do, we classify receptors into 4 types. Okay? We can classify them into 4 types. So please observe proper outlining ha. Balangkasin nyo na maayos yung dinidiscuss natin. Under ito lahat ng receptors. So first we have type 1 receptors. Names for type 1 muna tayo. Number 1, type 1 receptors are also known as ionotropic receptors or they are termed as ligand-gated ion channels. Okay? These receptors are said to be membranous, meaning to say you can see these receptors being embedded in the cell membrane. So this could be found in the cell membrane. And once a ligand, wag niyong kakalimutan ha, pag ligand, susi, kagaya ng gamot at natural compounds. Ang onset, pag sinabi nating onset, mangyayari ang effect, mangyayari ang cellular response pag type 1 na na-activate within milliseconds. So sobrang bilis. Pag dumikit yung messenger, pag dumikit yung gamot o dumikit yung compound sa active site ng type 1, within milliseconds may response agad sa cell. Okay? So yun yung characteristics ng type 1 receptors. So ngayon, i-explain natin ano ang characteristics ng type 1 receptors by referring in the figure. Are we still good? Okay pa ba tayo? May pumapasok pa sa utak? Ha? Okay, so I hope hindi pa tayo puno, hindi pa saturated yung nasa isip. Mag-health break tayo maya-maya lang, okay, para mapahinga yung utak natin. Okay, for example, let's have this illustration. As I've said, ang type 1 receptor ay membranous. Kaya you can see na ang type 1, etong nakabox yung binax ko, yan ang type 1 receptor. Okay? And then you can also observe that meron tayong active site where the ligand will bind. Now, provided that the messenger or the ligand has bound to the receptor, so it can cause opening of the channel. So initially closed siya, and pag bound na yung ligand, magbubukas yung receptor. Pag sinabi nating magbubukas, it will allow movement, okay? It will allow movement of ions. Okay? It will allow movement of ions. Yun yung function ng type 1 receptors. Ions ang pinapagalaw niya. Kaya ang tawag natin sa kanya ay ionotropic receptors. Okay? Ang tawag natin sa kanya ay ionotropic receptors kasi it can allow movement of ions whether papasok ng cell o kaya palabas ng cell. It depends in the situation. Okay? It depends in the situation. Okay? At depende kung anong ion ang involved. So yun yung tatandaan nyo in general sa type 1. Moving on. Sir, ano ang sense na malaman namin na naggalawan ang mga ions? Take note guys, pag naggalawan ang mga ions in and out of the cell, meron siyang epekto sa cell. It can make our cell stimulated or relaxed. It can make our cells depressed o kaya excited. Yun yung effect kapag naggalawan yung ions sa cells. So for example, because of the movement of activation of ionotropic receptor, pag naggalawan ng ions, and particularly pag dumami ang cation sa loob ng cell, ang tawag natin sa cell, depolarized na siya. Okay? Depolarized. In other words, excited ang cell. So kapag na-activate natin ang type 1 receptors at nag-dominate ang positive ions sa loob, that makes the cell excited or stimulated siya. Nagko-contract, active siya. Okay? Active. Yun yung effect ng pagdami ng cations due to ionotropic receptor. Uy, okay pa kayo? Okay. Next. Siyempre, hindi lang naman positive ions ang pwedeng gumalaw. Sabi ko nga, depende sa receptor na pinag-uusapan natin at ions. Meron namang mga receptors na pag na-activate sila, ang padadaanin nila sa butas nila mga anions. Kaya ang tendency, ang dadami naman mga negative ions or anions. That makes the cell hyperpolarized. Okay? That makes the cell hyperpolarized or depressed. Okay? That makes the cell eventually depressed. So in short, via type 1 receptors that can make the cell active o kaya relaxed or depressed. So yan ang type 1 receptors. Pwede na i-continue? Okay. So hindi pa tayo tapos overall sa type 1 receptors. Sir, paano namin ma-appreciate ang drugs dito? Okay, paano namin ma-appreciate ang drugs dito? Siyempre, magbigay tayo ng example na type 1 receptor. Ang example natin ng type 1 receptor ay yung tinatawag na GABA A receptor. Okay, again, ang example natin ng type 1 receptor ay ang GABA-A receptors. So for you to appreciate GABA-A receptors, i-explain muna natin ang kanyang function. Okay? Ang GABA-A receptors, kapag siya ay na-activate, hindi ako magaling mag-drawing nga, pasensya na. So kapag siya ay na-activate, halimbawa, ito yung GABA-A, at nag-bind ang isang ligand sa active site niya, ay nag-bind ang ligand sa active site, magbubukas ang channel ng GABA-A receptor. At pag nagbukas ito, gagalaw ang mga chloride ions papasok ng cell. Ay gagalaw ang mga chloride ions papasok sa cell. So intracellular, tumataas ang chloride ions. I have a question. An ion ba ang chloride? Negative ion ba ang chloride? Yes or no? An ion, very good. Therefore, pag na-activate ang GABA-A, anong effect? Depolarized or hyperpolarized? Hyper, very good. So that can cause hyperpolarization. Okay, that can cause hyperpolarization of cells, making the cell depressed. Okay, nare-relax niya ang cells. Okay, nare-relax niya ang cells. So yan po ang GABA-A receptors. Now, continue natin. Ang GABA-A receptor ay nagsilbi ring target para mag-discover ng gamot by medicinal chemists. So nakaisip sila ng pwedeng opportunity sa GABA-A receptors, particularly mag-discover ng gamot that could activate further GABA-A receptors. Kaya meron tayong gamot na nag-i-stimulate ng GABA-A receptors, ay nag-i-stimulate ng GABA-A receptors. At ang tawag doon mga benzodiazepines. Ang tawag doon ay benzodiazepines. Siguro mas familiar kayo sa specific example niyang diazepam. Narinig yun na yung diazepam? Valium, diazepam. Ang nakitang opportunity ng mga medicinal chemists dito is, pwede nating targetin ang GABA-A. Pwede tayo maghanap ng gamot na pwedeng mag-activate further ng GABA-A. So pag sinabing activate further, yung diniscuss kong mechanism ng GABA-A, sinusuportahan ng gamot. Kaya question, kung sinusuportahan ng gamot yung GABA-A activity, ano yung gamot? Ang gamot ba ay pampakalma o pampahyper? Pampa-stimulate ng cells? Pampakalma. Anong alam niyo sa diazepam? Ang diazepam ba ay sedative hypnotic? Ang diazepam ba ay pampakalmang gamot? Pampatulog ba siya? Sedative hypnotic ba siya? Yes. Kaya ang diazepam, as we all know, sedative hypnotic kasi GABA-A agonist siya or GABA-A activator siya. O yun, drug target siya. Gets? Nagets nyo? Okay. Next. Alright, let's continue. Introduce ko muna yung type 2 receptors bago tayo mag-health break. Okay lang ba? Introduce muna natin. Very good. So type 2 receptors tayo. After we finish discussing type 1, let's have type 2. Type 2 receptors are considered as the most abundant receptors in the body. Meaning to say, marami tayong mga receptors that are type 2. Kung marami tayong type 2 receptors, marami tayong potential targets na type 2. Kaya marami ding gamot available in the market. Type 2 receptors ang tina-target. Okay? These are also known as 7-transmembrane receptor, heptahelical receptor, and serpentine receptor. Pag sinabi nating 7-transmembrane up until serpentine, this talks about the structure of type 2 receptors. Okay? Pag hepta, 7. Kapag serpentine, mukhang ahas, di ba? Serpentine, serpent, ahas siya. Okay? So discuss muna natin yung second bullet. Bakit yan ang kanyang pangalan? By referring in the figure. So halimbawa, ito yung structure ng type 2 receptors. Ang type 2 receptors ay located din sa cell membrane, just like type 1. Okay? These receptors are also found in the cell membrane. And kaya siya tinawag na 7-trans membrane kasi pag tinignan mo ang type 2 receptors, 7 beses, beses siyang tumagos, 7 beses niyang tinuhog yung cell membrane. Kaya kung bibilangin natin, let's check kung 7 beses ba talaga. 1, 2, 3, 4, 5, 6, 7. Kaya siya heptahelical, mukha siyang helix, di ba? Kaya siya 7 transmembrane. And if you will check the structure, somehow nire-resemble niya or mukha siyang ahas, kaya serpentine receptor. Naintindihan po ba? Okay, very good. Next, doon tayo sa third bullet. Sa third bullet, tinatawag din siyang G-protein coupled receptor, and then we have metabotropic receptor. Bakit siya tinawag na G-protein coupled receptor at metabotropic receptor? Wag nyo muna tong picturean or wag nyo munang kopyahin, makinig muna sa akin. Okay? Kakadagdag ko lang nito kagabi, tsaka nyo na lang picturean or screenshot. So bakit siya tinawag na G-protein coupled receptor or GPCR, G-protein coupled receptor? Ang reason kaya siya tinawag na G-protein coupled, intindihin niyo mabuti ha, G-protein coupled receptor. Ito ay isang klase ng receptor na may attach, may link, may kadikit, may couple na G-protein. Kaya kung ito yung heptahelical structure ng receptor, may makikita tayong protein na naka-attach sa kanya. Ang tawag dito ay G-protein. Ang G-protein, discuss natin briefly, ang G-protein ay isang signal protein, ay isa itong example ng signaling protein, and meron siyang tatlong subunits. So these are alpha, beta, and gamma subunits. Again, we have 3 subunits. We have alpha, beta, gamma. Obvious naman sa picture. Alpha, beta, and also gamma. So yan muna yung introduce ko about G-protein. Signaling protein. Then it has 3 subunits. Kaya siya tinawag na G-protein coupled kadikit na receptor. Nagets yung term na GPCR? Okay. And then metabotropic, mamaya sa mechanism ko i-relate kung ba't siya tinawag na metabotropic receptor. Sa ngayon, yung G-protein coupled muna. Okay? So check tayo dito. Ito utang muna ha. Utang muna ito, yung metabotropic. Next, cell membrane. Okay na to kasi sabi ko sa cell membrane siya makikita. Onset seconds. Ibig sabihin, the message, the effect or cellular response is observed sa or within seconds. Question, mas matagal or mas mabilis compared sa type 1? Okay, slower, mas matagal. So right now, na-discuss natin ang general concepts about type 2 receptors. Kaya after our health break, i-discuss na natin yung picture na ito. So you can now screenshot this muna. O kung gusto nyo, buburahin ko pala ito mamaya. So screenshot nyo na ngayon. Okay? So pwede nyo na itong screenshot. And then, health break tayo. So kindly go back at 8:28 sana ready na kayo para 8:30 exact continue tayo. Naiintindihan ba yung lecture so far? Nice. So continue tayo at 8:30. Thank you guys. Thank you and see you later. Okay, so let's continue. After we described type 2 receptors, let's now proceed on the signal transduction mechanism involving them. Remember class that when we focus on GPCRs, like what I said, ito yung mga receptors na may kadikit na G-protein. So ano yung signaling mechanism na nangyayari involving them? And what's the importance of G-protein here? Being a signaling protein. So let's say for example, okay, by the way, bago ko mag-proceed dun sa mga steps, please bear in mind that may coupled GDP or guanosine diphosphate kay alpha subunit. Okay, kay alpha subunit. May importance ito that will be revealed later. Okay? So proceed tayo sa mga steps. Number one, suppose meron ng ligand binding. Okay? Suppose meron ng ligand binding sa active site ng GPCR. So in this regard, sa picture, yun yung agonist na tinatawag. Agonist is a type of a drug. Mamaya madidiscuss natin yon. So let's say there is an agonist or stimulator that has bound on the active site of GPCR. So pag nagkaroon ng ligand binding, that causes G protein activation. Okay, that can cause G protein activation. And when we say G protein activation, may mga certain processes tayong ma-observe once this G protein was activated. Number one, part of G protein activation is that the GDP, okay, GDP is being replaced. Okay, this is being replaced to GTP. So from guanosine diphosphate, the GDP is being replaced with triphosphate that is bound sa alpha subunit. Okay? And then part din ng G protein activation is that meron tayong dissociation of the subunits ng G protein. So meron tayong dissociation ng mga subunits. When we talk of dissociation, that simply means naghihiwalay. Okay, naghihiwalay, forming now dimer composed of beta and gamma subunit, a dimer composed of beta and gamma subunit, at monomer composed of alpha subunit. In Tagalog ng dissociation, naghiwalay yung tatlong subunits. Ang nangyari is nahiwalay ang alpha doon sa beta at gamma. Okay, ganun ang ibig sabihin. Gets nyo ba? Okay, so that's dissociation. All right. Now, kapag nag-dissociate na ang monomeric alpha subunit na may triphosphate, that can cause enzyme activation na. Okay? That can finally cause enzyme activation. So in short, yung prerequisite para magkaroon ng enzyme activation is kailangan mahiwalay ang alpha subunit na may guanosine triphosphate. Without it, walang enzyme activation. And what do we know about enzymes? Remember, nabanggit natin kanina, enzymes can participate sa chemical reaction forming now certain products. Okay? Forming now certain products. And this product is called a secondary messenger. Okay? Ang tawag natin sa product na nabubuo in general sa G protein activation ay secondary messenger. Ultimately, okay, I hope you can still follow, ultimately, this secondary messenger is the one responsible for the cellular response or effect. Okay? So finally, siya yung responsible sa cellular response or what you call as an effect. So yan ang mga general steps involved in GPCR activation. So brief review, didikit ang ligand, and then ma-activate ang G protein, magiging GTP, magkakaroon ng paghihiwalay, nakakasunod ba? And then yung monomer, yung solong alpha unit with the GTP, siya yung magsisilbing activator ng enzyme. Actually, para lang malinaw, depende sa G protein ha, may enzyme activation at may enzyme inhibition. Bago ko makalimutan. So pwedeng activation, pwedeng inhibition. Okay? Pero nag-focus tayo ngayon sa activation, forming now product that would cause a response. So yan ang mga steps ng G protein activation. So, if you can observe, kaya siya tinawag na metabotropic because, again, kaya siya tinawag na metabotropic kasi nai-induce din ang metabolism caused by enzyme. Okay? Nai-induce din ang metabolism caused by enzyme. And also, kaya ang G protein ay signaling protein kasi siya ang magsisignal sa enzyme na magtrabaho. Okay? Kaya signal protein siya. Gets? Hmm? Uy, pagod na ata ah. Ano, gets? Okay, nice. Siyempre, upang ma-appreciate ito, magbigay tayo ng sample application. Medyo ni-rearrange ko yung slide flow ha, pero meron din nito sa inyo. Bandang dulo ko lang ata nalagay. Okay, hanapin nyo na lang. Bandang dulo to. Pero under to ng type 2 receptors. Okay, under ito ng type 2. So let's have an example which are alpha 1 receptors. Which are alpha 1 receptors. By the way, bago ko makalimutan, guys, marami tayong klase ng G protein marami tayong klase. Pero pharmacology na ang bahala dyan. Okay, pharmacology. Madidiscuss sa inyo iba't ibang types ng G proteins. Okay, ako general process lang ang aking diniscuss. So going back, ang example natin for the application are the alpha 1 receptors. Alpha 1 receptors are G protein coupled receptors. Okay, so right now let's explain. Okay, let's explain what could happen once alpha 1 receptors are activated. O, imagine niyo ha, kung ang alpha 1 receptors ay isang GPCR, pag siya ay na-activate, magkakaroon ng G protein activation. Particularly, ang type ng G protein na na-activate kay alpha 1, Q kung tawagin, GQ. Okay, GQ. In which, pag na-activate ang GQ, magkakaroon nga ng replacement ng GTP mula sa GDP at hihiwalay sinong hihiwalay doon sa dalawa? Sinong hihiwalay? Alpha, beta o gamma? Alpha. Kaya makikita nyo sa picture natin, ang alpha subunit solo na siya at sino yung bilog na itim dito? Sino yung bilog na itim? GTP o D? Is it tri o di? Try. Kaya GTP ang nasa legend natin. Okay? Anong gagawin? Anong next step? Sinong pupuntahan nung alpha subunit? Pupunta ba siya sa enzyme? Yes or no? Pupuntahan niya ba yung enzyme? I-activate niya ba yung enzyme? Yes or no? Yes. So pupuntahan, tignan niyo mabuti nasa picture pupuntahan niya yung enzyme. Okay? Kaya sa next picture, makikita natin na yung alpha subunit with GTP, it bound to the enzyme called phospholipase. Okay? This can cause activation of the enzyme phospholipase C, okay, or PLC, in which this enzyme siya, so kaya niya mag-cause ng chemical reaction. This enzyme could convert PEEP2. This is the substrate for the enzyme. Okay, this is the substrate. Question, ano yung substrate? Reactant or product? R o P. Very good, reactant. So mapapansin natin, kanina, tingnan nyo kanina, nung hindi pa nakadikit ang alpha subunit, sarado yung pocket o yung active site niya. Pero nung nakadikit na yung alpha subunit, bukas na siya for PEEP2. Kaya mabubuo ang DG and IP3. Ano yung DG at IP3? Ang DG at IP3, yung products, syempre, yung products. Ito yung, anong tawag natin sa product pag type 2, anong tawag sa product? Secondary, ano yung M? Messenger. Okay. So secondary messenger. Ito ba yung responsible for the effect? Yes or no? Yes, okay, responsible for the effect. Okay? So yan yung mangyayari. So to specify this further, punta tayo sa next slide. So ito yon, okay? Kung ito yung PEEP2, again, kung ito yung phosphatidyl inositol or PEEP2, via PLC's action, may dalawang products, IP3 and DG, which are the secondary messengers. Okay? This IP3 and DAG sa loob ng cell can enhance activity or can enhance calcium inside the cell. Okay? It can enhance calcium inside our cell. And calcium is responsible for contraction of cells, like the muscles. Okay? Like the muscle. Okay? Contraction of cells like our muscle cells. So let's repeat. Ang effect ng IP3 at DG or DAG, that can cause enhanced calcium levels inside our cell, and calcium is responsible for contraction such as in the muscles. Okay? So, yan ang function ng alpha-1 receptors. In short, ang effect ng alpha-1 sa ating cells like muscles, contract. Okay, contract. So let's have an example drug in this case. Okay? Let's have an example drug. Nandyan pa kayo? Okay, example drug tayo. By the way, yung alpha-1 receptors na nagkokos ng contraction, highly concentrated ito sa ating blood vessels. Okay, highly concentrated ito sa ating blood vessels or ugat. So, pwede itong maging target, okay? Pwede itong maging target by medicinal chemists in discovering drugs. Kaya meron tayo mga gamot that can target alpha-1 receptors. Like for example, stimulators ng alpha-1 receptors. Okay, stimulators ng alpha-1 receptors. Question, pag stimulators ng alpha-1, anong ma-realize natin? Sila ba ay relaxant o sila ba ay constrictor drugs? Ano sila? Very good. They are constrictor kasi pag sinabi nating stimulator, yung pinaliwanag ko mula umpisa, ganyan din ang gagawin ng mga gamot. So yung ligand na stimulator, kung stimulator siya, lahat ng pinaliwanag ko hanggang dulo dito, ganun din ang gagawin ng gamot. Okay? Kaya constrictors sila. So example dyan, phenylephrine. Si phenylephrine ay isang alpha-1 agonist. Okay? Phenylephrine. Kaya si phenylephrine ay isang decongestant kasi vasoconstrictor siya. Okay? And then, we also have dilators. So kapag dilators, ibig sabihin, pinipigilan naman niya yung pathway na in-explain ko. Like for example, prazosin. Like for example, prazosin. Okay? So it gets tougher na, di ba? Pero basically, minomodify pa rin yung function. So yan po ang type 2 receptors. Actually, madaming type 2 receptors. So, may encounter niyo sa pharmacology ano pa yung mga iba't ibang types na or examples ng type 2 receptors. I'm just having some triggering information sa inyo kung paano ba nag-discover nga ng drugs. So finally, tapos na ang type 2 receptors. Next, punta naman tayo sa type 3 receptors. Ang type 3 receptors ay tinatawag na kinase-linked receptors or tyrosine kinase-linked. So isipin nyo, di ba, linked, parang G-protein coupled. Ibig sabihin, sa part na to, yung receptor may kadikit na kinase. Question, ang kinase ba ay enzyme? Yes or no? Yes, very good. So ang kinase, ano muna tayo ha? General concept muna para ma-breakdown natin mamaya. Ang kinase ay isang enzyme. Anong klaseng enzyme ang kinase? Ito ba ay, or anong klaseng enzyme ang kinase? Siya ay taga-transfer. Transferase enzyme siya. Okay, transferase. Meron siyang pinapasa, may tinatapon siya, may nililipat siyang functional group. Anong functional group nililipat ng kinase? Pag narinig ang kinase, ano nililipat? Tandaan nyo ha, mortal sin sa isang pharma student na hindi niya to alam. Kaya wag na wag niyo tong kakalimutan. Pag ang isang enzyme ay kinase, automatic, tinatransfer niya ang phosphate group. Okay, tinatransfer. Ililipat niya ang phosphate group doon sa substrate. So ang tendency, pag ang enzyme ay isang kinase, lalagyan niya, dadagdagan niya ng kinase yung kanyang reactant. Kaya ang action niya, phosphorylation. Okay? Nagets nyo ba? Gets? O yan na, mortal sin na yan next time pag hindi nyo pa matandaan. Kailangan alam nyo siya. Okay? Very important concept sa biochem and other pharma courses. So moving on, wag nyo kakalimutan kinase link. So dapat mababanggit natin ang salitang kinase mamaya. And the onset is in minutes. So ano napansin? Mas tumatagal ba siya? Mas matagal ba siya compared sa dalawa kanina? Yes, very good. So ang examples natin ay insulin receptors and growth factor receptors. Examples ng growth factor, we have epidermal growth factor. Search nyo na lang sa Google ha para mabilis tayo. So, nakasulat na pala sa slide, sorry. Epidermal growth factor and then platelet-derived growth factor receptors. Okay, so yan yung mga examples. So, paano natin maintindihan ang structure ng type 3? Bakit siya tinawag na kinase linked? Bigyan ko na lang kayo ng time mamaya to screenshot this, pero I just want you to visualize kung ano itsura ng type 3 receptors. Ang type 3 receptors is like this, okay? Or are like this. So ito yung type 3 receptors. So we have type 3 receptors found in the cell membrane. Actually, tagusan siya. Cell membrane hanggang intracellular part, hanggang sa loob. Pero generally speaking, cell membrane pa rin located. Okay? Bakit siya tinawag na tyrosine kinase? Linked receptors or kinase linked receptors. That is because, by the way, nakikinig pa sa akin? Nakikinig pa? Okay. So that is because when you take a look sa receptor, yung mga tyrosine residues, tyrosine residues ang tawag dito. Tyrosine residues, they could act potentially as kinases. So this tyrosine residues, siya ay makikita intracellular, tulog sila in the meantime. They are inactive domains yet. Okay? Inactive pa sila. Pero they have the ability to become kinases kapag sila ay nabuhay. Okay? Ano ang prerequisite para mabuhay sila, para ma-activate sila? That is a ligand from the outside. A key from the outside has to bind on the active site. So for example, epidermal growth factor, o kaya insulin, o kaya platelet-derived growth factor, they can bind on the active site. Okay? So, makinig guys, kapag nag-bind sa active site ang isang ligand for type 3 receptors, mabubuhay yung mga tyrosine residues sa loob. Pag sinabi kong mabubuhay, magiging active kinases sila. Okay, magiging active. Nagigets ba ako? Okay, nagigets ba ako? So, magiging active kinases sila. Pag sinabing active kinases, may kakayahan sila na magpasa ng phosphate. So this is how it goes. This is how it goes. Ang source ng phosphate ay adenosine triphosphate. Okay? Sa loob ng cell may ATP. Halimbawa, ATP to. Ilang phosphate meron sa adenosine triphosphate? Ilang phosphate? Tatlo. So halimbawa, 1, 2, 3. May tatlong phosphate. Okay? So may umaaligid na mga ATP, may umaaligid na ATP sa ating cytoplasm. So pag na-activate ang type 3 receptors, ito ay magsisilbing source ng phosphate by the tyrosine kinases na na-activate. So ang tendency, ang mangyayari dyan would be, okay, again, ang mangyayari dyan, kukuha ng phosphate group ang isang tyrosine residue at ipapasa niya sa kapitbahay niya. And then yung isa, papasa niya sa kapitbahay, papasa niya sa kapitbahay hanggang mapuno sila ng phosphate group. Kaya na-phosphorylate yung tyrosine residues. Kaya ang tawag doon, tyrosine kinase. O lalagyan kita ha, o bibigyan kita ha, kuha tayo kay ATP, lipat natin dito. So activated tyrosine kinases sila. Gets? Na-gets? Okay. So in the meantime, na-gets nyo ang term na tyrosine kinase. Pero wala pang effect, di ba? Wala pang effect. Mamaya pupunta tayo sa effect. One step at a time lang. O sige, screenshot nyo muna to. Then type done if you're finished. Okay, good. So dito tayo. Kakadagdag ko lang din ito. Mamaya screenshot nyo na lang din. So discuss na natin ano na yung mga specific steps na nangyayari sa tyrosine kinase receptor activation. Okay, paliwanag na natin yung step by step. So first question. Okay pa kayo? Mukhang module 2 lang matatapos ko today pala. Mukhang module 2. Okay. So, next, dito tayo sa step by step. So, una, let's say inactive pa siya. I-describe natin ang inactive form ng tyrosine kinase and linked receptor. Pag inactive pa ang type 3 receptors, meron tayong dalawang monomers na magkahiwalay. So first monomer and then second monomer. Magkahiwalay pa sila. And then you can see the tyrosine kinase domain here, a tyrosine kinase domain na inactive pa kasi wala pang ligand eh. Okay? So inactive pa siya. So yan ang first step. So magkahiwalay na monomers at meron silang tyrosine residues na inactive. Now, kung meron tayong ligand na nag-bind sa active site, that causes dimerization. Pag sinabing dimerization, magdidikit yung dalawang monomers. So yung una at yung pangalawa, nag-dimerize. Kaya may dimer tayong nabuo. May dimer. Nagdikit sila. Nagets niyo yung pagdikit? Ha? Okay. Continue natin. Kapag nagdikit sila, after they form a dimer, that can cause orange na yung kulay ng tyrosine kinase residue. Kaya ang tendency, activated na siya. Kaya kung aalalahanin niyo yung picture natin kanina, yung orange dyan, ito yon. Okay? Ito siya. Naging ganito na siya. Okay? Nagkaroon na ng phosphorylation sa bawat tyrosine kinase. Okay? Na-phosphorylate na sila. So what happens next? Yung orange, ito itsura ha. Next, number 3. Ay number 3, ito na yon. Phosphate, nakadikit na siya. So anong magiging role ng mga phosphate groups na dumikit sa kinase? Ang phosphate group na ito, this will serve as donors, okay, sila or sources, sila ay magsisilbing source para sa ibang mga proteins, tawag natin doon relay proteins to be activated. So in short, sa bawat phosphate group na dumikit doon sa tyrosine kinase, nagsisilbi siyang source para ma-phosphorylate yung ibang protein sa cell. Okay? So para ma-phosphorylate. So ganito siya kasimple. Pag nagkaroon ng phosphate yung kinase, ipapasa rin nila yon after sa iba pang protein sa loob ng cell. So pasa-pasa siya. Okay? Pasa-pasa. And overall, etong mga relay proteins, bawat isang relay protein na na-phosphorylate, na dagdagan ng phosphate group, may isang effect. Okay? May isang effect. So effect number 1 ito, effect number 1, tapos may isa pang relay protein na dumikit sa isang phosphate group. So may isa na namang effect, tapos may isa na namang relay protein, kaya may isa na namang effect. Kaya overall, ang realization nyo, pag type 3 receptors, grabe yung amplification of effect. Kasi bawat isang phosphate na ipinasa sa relay protein, may isang epekto, kaya multiple effects siya. Do I make things clear? Naintindihan nyo ba yung sinasabi ko? Okay. Gets? Sige, ulitin natin briefly ha. So tapos na yung type 3. Tapos na yung type 3. Okay? So yung bago nyo i-screenshot ito, i-recall ko lang briefly. So number 1, nakikinig pa sa recall natin. I-recall natin. Makinig ha. So first step, pag inactive, magkahiwalay yung dalawa. Next, pagdikit ng ligand, again, pagdikit ng ligand, ma-activate. Kaya yung mga kinases na to, kukuha sila ng phosphate group. Sino ang source ng phosphate group para magkaroon ng phosphate ang mga kinases? Saan manggagaling? Yes, ATP. Okay. Now, pag na-phosphorylate na ang mga kinases, question, ipapasa rin ba nila ito eventually? Yes or no? Yes. Sinong tatanggap? Anong tawag? Relay proteins. Very good. So bawat relay protein, isang phosphate. So phosphorylated yung relay protein, isang epekto. Pangalawang epekto. Pangatlong epekto. Kaya ang daming effects na kayang gawin. So yan po ang type 3 receptors. Okay? O screenshot nyo na. Sige. Type done if you are finished. Okay, nice. Next, magbigay tayo ng example ng type 3 receptor. Ito I think, meron kayo nito? Meron kayo nito? Parang nalagay ko siya, di ba? So, ang example natin ng type 3 receptor ay ang insulin receptor. Ang function ng insulin sa ating katawan is to lower blood glucose level, di ba? Hypoglycemic hormone to eh. Okay, that is to lower, again, that is to lower blood glucose level. So that's the function of insulin receptors. So ang mga mechanisms leading to low blood glucose level, multiple. Multiple kasi metabolic siya eh. Metabolic yung kaya niyang gawin eh. Ngayon, kahit papaano na a-appreciate natin bakit multiple ang effect ng insulin. Kasi type 3 receptor nga siya. Kasi pag meron tayong type 3 receptor, bawat phosphate may isang phosphorylation, may isang effect. Kaya amplified ang lowering effect ng lowering effect sa glucose brought about by insulin. So just like earlier, ganun din ang mangyayari. Insulin will bind on the active site causing dimerization, kaya magdidikit. Kaya ang mga kinases, maphosphorylate sila from ATP, and eventually that phosphate group will be transferred once more to different relay proteins, kaya merong further effects. Okay? Actually, kulang yung illustration kasi ang pinaliwanag lang dito hanggang phosphorylation lang ng kinases. Di ba? Di ba kinase ito? So nalagyan sila ng phosphate groups due to phosphorylation from ATP. Yan. So overall, tapos na tayo sa type 3 receptors. So finally, we have type 4 receptors. Type 4 receptors are also known as gene transcription linked receptors. Question, anong ibig sabihin ng transcription? Anong pinoproduce mo pag transcription? Anong ginagawa sa katawan kapag transcription? Protein ba ang ginagawa or RNA? RNA, that's right. So RNA. RNA ang pinoproduce kapag transcription. Pag translation ang protein. Okay? On the contrary, from the first three types, ang type 4 ay nucleus located. Nucleus. That's the reason why in other references, ang tawag sa kanya ay nuclear receptors or intracellular kasi nasa loob ang ating nucleus. And siya ang pinakamatagal in terms of effect kasi in terms of hours siya, ay in terms of hours. So examples are steroid hormone receptor, thyroid hormone receptor, vitamin D and vitamin D derivatives. So just to show you briefly kung ano nangyayari sa type 4 receptor, ganito siya. Remember that type 4 receptor is found inside the cell. Kaya this requires entry of the ligand. Again, makinig. It requires entry of the messenger or the ligand inside the cell. Okay? Tatawid siya. So after entry, of course, it will bind on the active site of the type 4 receptor. Okay? It will bind on the active site causing dimerization then. Okay? Dimerization. And after dimerization, via the participation of a co-activator protein, that would finally cause detection of the gene from the DNA. Pag sinabi nating detection of the gene, itetrace kung anong part ng DNA ang gagawing RNA. Kaya from this part, pag nakuha yung specific gene from the DNA, ay pag nakuha yung specific gene from the DNA, yun na yung magiging transcript or yun na yung magiging blueprint rather, okay? Template para makagawa ng RNA. Kaya gene transcription link siya. Ay yun yung nangyayari dyan. Limited lang yung mga, hindi ganun ka-common yung mga drugs sa target type 4. Unlike type 2 and type 1. Ay, marami kasi sa type 2 at type 1. Okay? So taken all together, that's the end of types of receptors. So natapos natin ang mga types of receptors and natapos natin yung mga targets ng drugs overall. So brief review tayo. Please answer what is being asked. Okay? Sa receptors, ilang types ang na-discuss natin? 4. Very good. Okay, 4. Sa apat na receptors, sino ang naiiba sa location? Sino ang naiiba sa sa receptor location? 1, 2, 3, or 4? Very good, 4. Sinong fastest? Very good. Sino ang kinase linked? 3. Abundant. 2. Sino ang nag-aalaw ng ion movement? Ion movement. 1. Sino ang G-protein linked? 2. Sino ang nagpo-produce ng RNA? Sino ang insulin or ano ang insulin receptor? 3. Sino ang mukhang ahas, serpentine type? 2. Very good. Ay, very good. Ano pa ba pwede ko itanong? Wala na akong matanong. Last question. Wala na pala. Wala na akong maisip. May naisip ako kanina, nakalimutan ko lang eh. Ayun. Sino ang naka-base ang activity sa GTP? GTP. 2. Very good. 2. Okay. GTP. Last na talaga to. Last na, promise. Amplification of effects and phosphorylation. Yes, type 3. Gets ba? Natututo ba? Okay. Review tayo ulit. Review. So far, ang natapos natin, yung mga types ng proteins targeted by drugs. Generally speaking, meron tayong 4 na na-discuss. Tama, 4. Number 1, structural. Nagigets yung flow? Structural. Number 2, T. Ano yung T kanina? Ano yung T kanina? T. Yung proton pump. Transport, yes. Alpha-glucosidase 3. Ano yung pangatlo na alpha-glucosidase? Ano yon? Ano yung pangatlo? Alpha-glucosidase. Ano yung alpha-glucosidase? Days. Ano siya? Ano yung pangatlo? Enzyme. Yes, enzyme. And then yung maarte that accepts ligand in lock and key form? Receptor. Yun yung 4. Tapos subtype na ng receptor yun. Kaya pag nagbalangkas kayo or nag-outline, guided kayo sa lecture. Okay? So next, another lesson. Actually sa module 2, 2 lessons eh. Lesson 1 yung types ng protein and then lesson 2 na ito. Okay, lesson 2. So proceed tayo ngayon sa drug-target interactions. So in this part, we have to identify the concepts that we should consider para ang gamot ay makapag-interact properly with the target protein, whether it's a receptor, an enzyme, a transport protein, or structural protein. Okay? We have certain considerations. So key concept, the interaction between a drug and a target, again, the interaction between a drug and a target is called as binding. So common sense, binding ang tawag doon. Now, yung term natin dun sa pocket ng target, again, ang tawag natin dun sa pocket na meron ang target na pupuntahan ng drug, binding site. Okay, binding site. Now, ano yung important consideration na dapat nating malaman? Important dito is that ang gamot dapat at ang binding site by the target, they must have a complementary interaction. Pag sinabi nating complementary, dapat nagmamatch sila ng interaction sa isa't isa for them to be compatible with one another. Okay? So ang determinant para maging complementary sila, forces of attraction, yung chemical bonds na makikita natin. Often, often, ang chemical bonds na nag-exist between the drug and the target, weak forces lang sila. Generally, ang tawag doon ay intermolecular forces of attraction. Okay? Intermolecular forces. So siya ang nagsisilbing puwersa para magkaroon ng compatibility ang drug at ang target. So for example, in this slide, kung ito yung macromolecular target having the binding site, of course, yung gamot, it must have certain functional groups na magkakaroon ng compatibility doon sa target that would cause attraction with one another. And the forces that would attract between the drug and the binding site are the intermolecular bonds. Okay, intermolecular bonds. Are these bonds weak or strong, generally speaking? Weak. Okay, weak. Okay, weak forces. I'll explain later bakit dapat ang consideration natin ay weak lang, not strong. Okay, I'll explain later. So, mamaya ko explain itong example, sa dulo ko siya explain. So for now, dito muna tayo sa mga intermolecular bonds. Talking about intermolecular bonds, these include weak forces of attraction like ionic bond, uh, van der Waals forces of attraction, and also hydrogen bonds. So isa-isayin natin sila. Briefly, number one, ionic bond. Ang ionic bond, ito rin yung electrostatic forces that involve interaction between oppositely charged particles or groups. So when we say oppositely charged, may ionic bond between a positively charged functional group or portion ng structure and negatively charged. Ganun lang siya kasimple, okay? And then this is considered to be weaker than covalent. Covalent will be discussed later, and distance dependent. So this will happen if there is close proximity between two oppositely charged groups. So that's ionic bond. Next, hydrogen bond. Basically, hydrogen bond is being formed between hydrogen atom and an electronegative atom. Electronegative atom are the ones that are found in the right side of the periodic table, like fluorine, oxygen, and also nitrogen. Just like ionic bond, ang hydrogen bond ay weak force din. So intermolecular bond din siya and reversible din siya. And finally, we have hydrophobic. Pag hydrophobic or van der Waals or London dispersion forces, by the way, pag FA, forces of attraction ang ibig sabihin yan, ang point ng hydrophobic, ito naman, between nonpolar bonds. Ay, between nonpolar. So kung babalikan natin, pag ionic, oppositely charged. Pag hydrogen bond, hydrogen plus electronegative, such as FON. And then hydrophobic, non-polar groups. Yan ang mga intermolecular bonds na kailangan nating i-consider. So like what I said kanina, i-discuss ko yung example bandang huli. So ngayon na natin siya i-discuss. So para maintindihan nyo lalo yung ibig sabihin at importansya ng intermolecular bonds, let's have this example. Of course, ang priority natin na pag-aaralan is the receptor kasi this is already God-given. Ito na talaga yung bigay ng Panginoon, ito na yung structure niya. The one that we should adjust, yung gamot. Okay? Kaya unahin natin ang receptor. Of course, we shall focus on the binding site of the receptor. Anong mga functional groups ang makikita sa binding site? Okay? So for example, this is the binding site of the receptor, and hypothetical, it was determined that at this given receptor, meron tayo makikitang OH groups, okay? OH groups, and then we also have this nonpolar region, and then we have negatively charged group. Again, we have tatlong areas ang nakita sa binding site. Number one, we have the nonpolar, a nonpolar region, a nonpolar. And then, yung region na may OH group and also negatively charged region. So ano ang dapat nating i-consider? Para ma-attract yung i-design na drug dito, dapat ma-establish natin ang mga forces sa binding site like hydrogen bond, like van der Waals, and ionic binding or bonding, ionic bonding. So in this regard, pag nag-design tayo ng gamot, dapat ang i-design nating drug ay complementary doon sa group na meron ang binding site. Are you getting me? Naiintindihan ako? Hmm? Okay, para magkaroon ng attraction. So kaya pa magde-design ng gamot upang makadikit doon sa binding site, okay, dapat yung gamot meron ding non-polar region. Kaya important ang organic chemistry dito. Merong benzene ring. Question, lipophilic ba ang benzene ring? Hydrocarbon siya. So puro carbon at hydrogen. So non-polar ito, okay? Non-polar. Nakikinig pa ba sa akin? Non-polar ang benzene ring, okay? Non-polar siya. And also we have the OH group. May OH group din yung drug na dinedesign or dinesign OH group. So may electronegative atom tayong oxygen. So may nakikita ba tayong opportunity kapag may electronegative yung structure ng gamot na dinedesign? May prospect ba tayong intermolecular bond na magbubuo pag may OH? Yes, meron. So pangalawa tong binding group na pwede mag-complement. And then in this given structure, meron tayong charged nitrogen. Anong charge na nitrogen in this given structure? Positive or negative? Positive. Very good. Positive. So, yung dinesign na gamot, yung pinakitang gamot dito, complementary siya. Okay? Complementary. Kaya, we can predict actually that it could fit on the binding site. So, makikita nyo dito na ang ina-adjust ang minomodified drug kasi receptor yun na talaga siya. Kaya pag pinakita natin dito yung attachment ng drug doon sa binding site, yung aromatic ring na non-polar, pumunta siya sa non-polar na region. Kaya nag-bind siya. Question, anong puwersa ang nag-e-exist sa dalawang ito? Anong puwersa ang nag-e-exist? H, van der, o ano isa? Ionic. Yes, van der Waals. Okay, next. Dito, bakit nagkaroon ng attachment dito? Merong, yung susunod, etong binilugan ko, anong meron dito? Electronegative, yes, hydrogen bond. Nice. And then, dito, positive, negative, anong tawag dito? Nice, ionic. Kaya, nag-attach siya. Okay, na-attach siya. So, yan ang importance ng chemical structure ng gamot. Kaya nga sa medicinal chemistry, we keep on developing and designing drugs in terms of their chemistry so as to establish intermolecular bonds promoting complementarity. Okay, doon sa binding site ng target at ng gamot. Okay, at ng gamot. Guys, promise, na-gets nyo ba? Hmm? Okay, so very good. And yan yung example natin, hypothetical example. Pero let me tell you guys that although majority ng drugs ay intermolecular bonds ang kayang gawin, syempre may exception to the rule pa rin. Meron tayo mga gamot na ang kayang gawin covalent bond. Okay, covalent bond. So para magets ito, i-describe muna natin ang covalent bond. Sa lahat ng forces of attraction between the target and the drug, ang strongest ay covalent. Okay? Pag sinabi nating strongest, that means pag nagdikit ang gamot at ang target ng covalent, permanent na siya. Sa buong lifespan nung tinarget na protein, hanggat buhay yung protein, nandun yung gamot. Kaya long-term yung effect niya. Kaya we can conclude that covalent bond is irreversible. So yung mga gamot na maririnig nyo, this drug is an irreversible agent. This drug is an irreversible inhibitor. This drug irreversibly blocks. Yung mga term na irreversible, covalent bond yun agad. Naintindihan nyo? Ha? Okay. And pag irreversible, tandaan na, di ba pag mas matagal kang nakadikit, mas mataas ang chance na matoxic ka. Pag mas mataas ang chance ng attachment, mas mataas ang chance na may toxicity doon. So dito, usually kapag ang gamot ay nag induce ng covalent bond sa target, highly toxic yung drug. Example, we have phenoxybenzamine, which is an irreversible alpha blocker, and then organophosphates, which are irreversible anticholinesterases. So these are irreversible agents. So nabanggit natin, hindi sila ganun ka-common compared doon sa mga reversible. And furthermore, kagaya ng sinabi ko kanina, ang intermolecular bonds, weak sila, so reversible sila. Irreversible. So yun po ang covalent and also intermolecular bonds. Class, na gets? Hmm? Okay, nice. O, last slide na to for today. Yes, finally. Module 2 lang pala matatapos ko ngayon. Okay, so last slide na to for today. Pen and board to, hindi ko nagawa ng PowerPoint, sorry. So discuss natin ang classifications ng ligands. Okay, classifications of ligands. So meron tayong classifications of ligands. So number one, meron tayong tinatawag na agonist. Ay meron tayong tinatawag na agonist. And then number two, meron tayong tinatawag na antagonist. So I'm referring here to the drugs na ha, to the drugs. So I repeat, we have two classifications of ligands. These are the agonist and then the antagonist. So ito yung dalawang classes. So bago ko i-discuss yung characteristics ng agonist at antagonist, meron lang ako i-define dito. Okay, meron lang ako i-define sa baba. Meron tayong terms called affinity, affinity, and then intrinsic, and then intrinsic activity. So we have the terms affinity and also intrinsic activity. So number one, when we say affinity, this is the ability of the drug to bind, okay, to bind to receptors. Again, when we say affinity, this is the ability of the drug to bind to receptors. Whereas intrinsic activity is the ability of the drug to activate the receptor. Again, when we say intrinsic activity naman, this is the ability of the drug to activate the receptor. Type done if you're finished copying so that I can continue. Done? Okay, very good. Anong ibig sabihin ng affinity? Pag sinabing affinity, kakayahang ma-attract, kakayahang dumikit nung gamot doon sa receptor. So this can be achieved by modifying the chemical structure of the drug para maging complementary siya doon sa binding site ng receptor o ng protein. Okay? Pero nabanggit natin kahapon kung natatandaan nyo, may mga gamot na or para malaman natin na effective yung gamot, syempre, dapat yung gamot powerful enough na mag-cause ng effect. Okay? Intrinsic activity naman yon. So yan yung difference. So based on this information, madi-differentiate natin ang agonist at antagonist. Ang agonist, para siyang activator, yun ang ibig sabihin, kasi ito yung klase ng gamot na mayroong affinity at intrinsic activity. Okay? So this is a drug that possesses both affinity and intrinsic activity. Whereas ang antagonist, it has an affinity but without intrinsic activity. Okay? So we have agonist, affinity plus intrinsic activity, and then antagonist, affinity but without IA. So yun yung difference ng agonist at antagonist. And when we focus on agonist, meron tayong full, partial, at inverse agonist. Ay meron tayong full, partial at inverse agonist. Kapag full agonist, ang kanyang IA or intrinsic activity is equal to 1. Again, ang kanyang IA is equal to 1, meaning 100%. Ang partial agonist, ang kanyang intrinsic activity is more than 0 but less than 1. Okay, but less than 1. Again, intrinsic activity is more than 0 but less than 1. At ang inverse, intrinsic activity is less than 0. Full, IA is equal to 1. Partial, IA is more than 0 but less than 1. Inverse, less than 0. Type done if you're finished copying so that we can summarize all the things that we discussed. Okay, very good. So overall, mula kaninang umaga until now, that ends Module 2, Drug Targets. So tapos na yung discussion natin. Stop share na ako, no?